Cette conférence va maintenant être enregistrée.
Good evening, everybody. Assalamu alaikum. So it's already time for our third session of this webinar on an introduction to laboratory design for biosafety purposes. This session will be completely devoted to answering the questions you asked during the two first sessions, and you posted in the chat box. There were two main topics that were covered by your questions. The first one were the first questions were related to the space organization of the lab, so mainly architectural layout aspects. So there were 8 questions there, and then some others were related to more technical aspects, exclusively on ventilation and air conditioning. There were also 2 additional questions, but that were out of scope. They were mostly dealing with risk assessment and not really totally to biosafety. So I won't answer those 2. So the layout aspect. First, as an introduction, space organization of the lab is not something that has to be done by one person. It's really teamwork. There has to be a number of a kind of project team around a project leader that deals with laboratory design, and there should be laboratory users, some high-level staff members, but also architects and engineers, the biosafety officer, and so on. So it's really teamwork because there are many different aspects which are related. So let's go to the questions now. The first question was, is it okay to use minimum area of lab as a workstation to restrict movement, or should we use separate labs for each activity? I was wondering what was exactly meant by workstation, because a lab is actually a number of workstations. So I suppose that it's a write-up area and a part of the lab which is used mostly for some office work. So the general principle is still to try to segregate activities based on their nature and risk. The rationale for that, the reason for that, is that we want to avoid potentially exposing staff which do not need to be exposed because they are not working on this. But we also need to take operational needs into consideration. So if it's a low-level laboratory, for instance, and people always have to go in and out to treat their results, it's probably not the best thing from an operational need. So since a write-up area is a no-risk area, and labs are places where there are some activities which can generate some risk, we need to base the decision on risk assessment on one side and also on practical considerations. In practice, there are 2 possible decisions. If the laboratory activities are considered low-risk activities, so there is a limited risk of exposure, and the consequence of an exposure would be minor. And if it's the same staff that uses the write-up area than those who work in the lab, in that case, it's possible to install a write-up area in the lab itself. It's possible to do it, but it might also be more convenient, and so it could be a good thing. We should take some particular precautions with respect to location, rules, and so on, and we'll come back on that immediately. The other possible decision is if you consider that the laboratory activities are high-risk activities, where, for instance, you would need more PPE than the basic lab coat and gloves, for instance. In that case, clearly, the write-up area should preferably be installed outside of the lab. What precautions to take when we have the write-up area in the lab? You should first locate it in the lowest risk area, if possible, too close to the exit door, and not close to the high-risk area. Those people, in case there would be a spill in the high-risk area, those people should not be directly exposed, and that should have the time to leave the lab without any problem. Also, you need to define rules about that. Should people who work at the write-up area, should they keep their lab coat or not? That has to be defined clearly. Also, that write-up area is not for other office work. It's just to treat the data that are produced in the lab, just to treat information that is in direct relation to laboratory. So it's not the place to do some internet search or anything. It's not, of course, not a place for eating and drinking, and not a place for meetings. So it should really be restricted to doing some office work in direct relation to the data that are collected in the lab. Another question about location: where should a molecular biology lab be located in a multidisciplinary laboratory unit? Actually, molecular biology is quite wide, and there are different types of activities there. I put there on the slide the three main ones that I see. The first activity could be work on E. coli or some other non-pathogenic organisms that are genetically modified and together with their plasmids. In that case, there is no biological risk and there is no particular issue. I mean, no specific issue related to contamination, for instance. Another activity could be work on viral vectors like antiviruses, for instance. And in that case, there is a low level biological risk. Mainly, the main risk would be in case of accidental injection, but there is also a need to protect at least the cells that are used for the viral culture. So you see that it's already quite different activity. And then a third major activity in molecular biology labs would be the handling of nucleic acids that like DNA and RNA. For instance, for PCR activities, which are now in most labs. In that case, again, there is no biological risk, but we need to protect the samples from any type of contamination, at least for the first steps of PCR. I will develop that a little bit more. So, if it is possible, the best thing to do would be to separate such activities from each other. So not do that all that in the same lab, but also from other activities of different nature and level of risk. It will also allow you to better ensure product protection and avoid any type of contamination. So that's really the best practice, but it's not always possible. And so in that case, if there are low level risk, biological risk of no biological risk activities, they could be done in a general lab. Brief question on PCR. Since it's becoming the main molecular biology activities in many labs, in diagnostics and also in R&D, I will share some best laboratory practices related to PCR. There is a microphone which is not on mute, I think. Please turn it off. Actually, if we look at PCR, it's one activity, but which is made of different processes, different steps, and each of those steps presents some different issues. The first part, you need to extract the DNA or the RNA. In that case, there is generally a biological risk, since the extraction is generally made from some infectious materials. And that biological risk will stay there until the first steps of cellular lysis. In that case, you destroy the cells, and then after that, there is no biological risk anymore. And there is also no major contamination issue, at least if you use good microbiological practices. Okay, that's the first process. The second process would be the preparation of the master mix and the sample, and that's what we call pre-PCR. There is no biological risk there. It's a chemical activity with low chemical risk also, but it's really extremely important to avoid any contamination of your sample. If you contaminate your sample, you will have false positive or you will be unable to read anything after the PCR. So it's really something which is extremely important to avoid any contamination. Another part of the process is the PCR itself, which is the amplification of the nucleic acid and the reading. Again, no biological risk and no contamination issue because it's done in closed tubes. And then after that, most of the time, you will use your amplified DNA or RNA to do some further analysis on that. And that's what we call post-PCR activities. So with respect to laboratory design, the best practice would be to separate all that. First, the extraction. If it is from some living materials, from infectious materials, at least the first step, the cellular lysis, should be done in the cell culture lab, where there are some biosafety practices in place. In that case, you avoid to bring any biosafety issues in the other labs. Okay? Then the pre-PCR, that's the the crucial activity where you cannot have any contamination. It should be in a small, dedicated lab. That lab should be totally separate from the extraction, if possible, but mostly from the PCR itself. And in that lab, you should have really high-level product protection practices in place. Then the PCR itself, there is no biological risk, no risk of contamination. It can be in another lab, which can be a general lab or it can be a small PCR. And post-PCR activities, it could be in the PCR lab or possibly in some general analytic lab. So there you have some flexibility. So in conclusion, depending on your infrastructure, depending on what is possible in your facilities, it should ideally be in 2 or 3 labs, beside the culture lab used for the extraction. I took an example of a PCR facility which is made of 2 labs. In that case, you have the pre-PCR in one lab, and there you only do the pre-PCR. You only prepare the master mix, and so you have a PCR workstation for that, and you have some other equipment. You don't necessarily need a sink or anything. In the second lab, you do the PCR and also all the post-PCR. And in that case, you have different, you can have a thing, and you have the PCR instrument, but also some equipment for the analysis, like electrophoresis, imaging, gel imaging systems, and so on. Okay? What is important is to have a flow of the samples from pre-PCR then to PCR and post-PCR. Also, reagents and so on should only go that way, from pre-PCR to PCR, and never the opposite. This should also be applied to the people. The people who work in the pre-PCR lab can then work in the PCR lab, but not the opposite. They should not go back into the pre-PCR lab. So it requires some organization there. However, in some situations, mainly existing labs, there are not enough rooms available for this. And in that case, if it's not possible to apply the best possible practice, in that case, you could conduct all activities in the same lab. But that has to be done with some well-defined precautions. Of course, you should use different benches in order to avoid any cross-contamination, and they should be located as far as possible from each other. Also, you should have different pieces of equipment from the pre-PCR on one side and for the PCR and post-PCR on the other side. You should also make sure that there is one direction workflow from B to C and D, and that materials and reagents that are used in C or D should never go back to B, should never go back to the pre-PCR area. As said before, it's also true for the personnel, staff, technicians and so on who have worked in PCR and post-PCR should not go back to pre-PCR during the same day. And if you use some ventilation system, in that case, you should also use the directional airflow from clean pre-PCR to contaminate C and D, so to PCR and post-PCR. Okay, so even with respect to ventilation, you should use that in order to avoid any contamination of your premix. Another question was about the trade-off between biological hazard and chemical hazard while designing a lab, and that's very important. And it's a question that is raised most of the time for different types of activities. The general answer would be to base decision again on risk assessment. You need to do the risk assessment for biological activities and for chemical activities. In practice, if possible, if the work process allow it, and if you have enough space and equipment to do it, in that case, you should apply segregation principles and plan for high-risk biological activities in one lab and for high-risk chemical activities in the other lab. It's not always possible. Sometimes you don't have a possibility to do that. In that case, you need to kind of weight biological versus chemical risk. If doing both risk assessment, you consider that the biological risk is more important than the chemical risk, in that case, you should design your lab according to the biosafety principles. And then when it's done, you should look at the situation with respect to chemicals and add possible additional measures that would be needed to control chemical risk. If the chemical risk is judged is deemed more important than biological risk, in that case, you should design your lab as a chemical lab, and then look at what you should need to have as additional measures to control biological risk in a good way. Okay. The next question was in relation with this one: what to do with respect to biosafety cabinets or fume hood, considering space and financial constraints. It's true that those two types of equipment, biosafety cabinets and fume hoods, cost quite a lot of money, and they also require some space. Okay. The first thing to do is to think about the basics of both types of equipment. Biosafety cabinets are made to protect people from solid and liquid aerosols. It's mainly liquid aerosols because we mainly deal with liquids in biological labs. And so they will, the filters will trap those droplets that may contain biological agents, but also dissolve chemical substances. So chemicals which are dissolved in your liquids will also be stopped by the filter. On the other hand, if your toxic substances are volatile, if they are in the air and not in droplet, in that case, they will pass through the filter, and so you could be exposed. Fume hoods are designed to protect from chemical substances, including volatile substances that could possibly be trapped on charcoal filters. So you have the exit of the fume hood. If you connect it to a duct that sends the air to the outside, it will also evacuate aerosols. It will not necessarily stop the aerosol through a filter, but at least you will you will eliminate the aerosols from the lab. So it will also offer some protection of the people working in the lab from those aerosols. So if you don't have enough space or enough money to get the two types of equipment when you would need it, you should decide about one or the other. The choice should again be based on respective biological and chemical risk assessment. But in that risk assessment, you should also consider the volume of the liquids you will use. If you use volumes of chemicals and only microliters of biological agents, in that case, it's probably better to give priority to chemical risk. Also the frequency. If most of the time you work on biological agents and that once in a while you need a little bit of toxic chemical that are, in that case, you should give priority to biological risk and then mostly have a biosafety cabinet. Anyway, if you try to manage both chemical and biological risk in the same protective equipment, in that case, the best options would be a hard ducted, so ducted to the outside, class 1 biosafety cabinet or fume hood. There is only one situation where you could prefer a class 2 biosafety cabinet is if the product protection is as essential as the personal protection. In that case, you could have a class 2 biosafety cabinet, but in that case, you would have a less good protection versus the biological risk and versus the chemical risk. Another question was, don't we need a full mirror? The first thing I would like to say is that a mirror is not needed. It's nice to have, it's not a core requirement, but it's also not expensive. So if it's useful, why not? If actually a mirror would be mainly useful to adjust things like a respiratory mask or like a head cover. And for that, you don't need a full-length mirror. You would only need a full-length mirror if you have a kind of overgrowing like this one. And this is not done in most labs. That's only for some very high-risk laboratories or in some GMP, good manufacturing production facilities, like in vaccine production, for instance. But anyway, all those are height control measures, and you should really adapt your decision due to the situation. But in most cases, a small mirror should be enough. I think it's the last question of this section. No, it's not the last. Should spill kit be located in the lab or in the entry room? And also, what should be the exact location of a spill kit in the lab? Actually, there was the start of a debate in the chat box about it. Again, like almost always, the answer is in the risk assessment. But before getting a little bit deeper into that, I want to give you some consideration. The first thing is that, do we really need a spill kit? Actually, in most cases, you won't need a spill kit. A spill kit is only needed if you handle large quantities of liquid that are potentially hazardous. If you use small quantities like microliters, milliliters, and so on, and that you always absorbing materials and disinfectant in the lab, which should be the case, in that case, you don't need a spill kit. So a spill kit is not a core requirement. Actually, in the risk assessment, you should have two specific questions. What immediate emergency measures would you put in place in case of a major spill? And then the second question would be the procedure. What would be the procedure to clean up the spill? How would you proceed to clean up the spill? And then if you need a spill kit, so for large volumes and so on, in that case, you also need to decide about the size of the spill kit, how much volume of absorbing material, and what will be the exact content. You will need also some specific PPE into your spill kit, for instance. So let's go to the risk assessment. In high-risk situations, that means where you handle some infectious liquids or some chemical hazardous liquids in large volumes, that means in most cases where you would need a spill kit, the ideal way to proceed in case of a major spill would be first to evacuate the lab. So everybody leaves the lab immediately. Why to do that? Because you want to minimize the risk of exposure to the aerosols or to the chemicals. The best way to do is to get out of the lab as soon as possible. And you leave the spill there. Okay? Then the second thing to do is only re-enter the lab for the cleaning up after some time. Why after some time? Because you need to let the aerosol settle on the floor, and that will be for the largest particles, or be exhausted through the ventilation system, and that's for the finest particles. And that time could be like, for instance, 30 minutes. That's if you have a reasonably good ventilation system, 30 minutes should be enough. But you could also wait for a longer time, okay? So in this case, since you have to evacuate the lab and only go back to the lab after some time, like 30 minutes, the best possible location of a spill kit would be outside of the lab and not in the lab. If you have an entry room, it could be in the entry room, and anyway, it could also be in the corridor. And in the corridor, I see one major advantage is that you can share a spill kit between several labs. If you have different labs which have different types of biological activities that handle large volumes, for instance, if you have the spill kit in the corridor, each lab could possibly use it. Remember that a spill kit would only be used exceptionally, like if it's really exceptional that we have a major spill. So it could easily be shared. Another question was about, is it acceptable to keep consumables such as reagent kits in the entry room? My point of view there is that an entry room should only be used as an entry room and not for the storage of things that should not be there. And certainly not if those consumables like reagents that present some hazard. If it's used for other activities like storage or anything else, then it will lose the entry room will lose its role of safe buffer zone. So when you design a facility, you really need to define the size of the entry room so that it best corresponds to its main functions, like donning and doffing and hand washing, and also the storage, that everything that goes together with this, like the disposal that are needed for that, lab coats, PPE, but also soap, for instance, for hand washing. And you could also store a spill kit and the cleaning materials, because it's better there than in a lab. But you should not make it so large that people are tempted to leave other things there. So, Madam Chada, could you please summarize this first part in Urdu?
Can you see my screen?
Yes, ma'am, please continue.
Okay, thank you so much. میں شاہدہ ممتاز قریشی ہوں اور میں آج اس سیشن کے لیے لیب ڈیزائن ویبنار کے جو سوالات اور جوابات ہیں اس کا سیشن آپ لوگوں کے لیے منعقد ہوا ہے. تو پہلا سوال جو کیا گیا ہے یہ لیبارٹری ڈیزائن کے بارے میں کل جمع 8 سوالات جو کیے گئے ہیں وہ اسپیس آرگنائزیشن کے اوپر ہیں کہ اسپیس لیب کی کس طرح سے ہونی چاہیے لیب ڈیزائن میں اور لے آؤٹ کیا ہوتا ہے, اور جو 5 سوالات ہیں وہ ہوا کا کس قسم کی ہوا اور ایئر کنڈیشننگ وغیرہ جو ہے وہ لیب میں ہونی چاہیے اس کے بارے میں ہے. تو لیب جو ہے یہ ایک مجموعہ لوگوں کو مل کر لیب ڈیزائن کے اوپر بات کرنی چاہیے. پہلا سوال جو کیا گیا ہے وہ لوگوں نے پوچھا ہے کہ کیا یہ صحیح ہے کہ ہم لیب کے اندر کوئی رائٹنگ ایریا مقرر کیا جا سکتا ہے. اگین آپ لوگوں کو دوبارہ سے اپنے کام کی تشخیص کرنی پڑے گی کہ کس نوعیت کا کام آپ لوگ کر رہے ہیں. اگر کام جراثیم کی ہینڈلنگ کا زیادہ ہو رہا ہے, اس کی نمونے کی تشخیص آپ زیادہ کر رہے ہیں, تو پھر ایسی لیب کے اندر آپ کوئی لکھت پڑھت کا کام نہیں کر سکتے بلکہ لیب کے باہر کرنا پڑے گا آپ کو کام. تو بہتر ہے کہ آپ پہلے تشخیص کر لیں. ہاں اگر کام ایسے جراثیم سے ہو رہا ہے جو زیادہ آلودگی یا کنٹامینیشن نہ پھیلا رہے ہوں تو ممکن ہے کہ آپ کچھ علاقہ لیب کا مقرر کیا جا سکتا ہے. لیب کا عموماً جو پہلا حصہ ہوتا ہے لیب کا اس میں ہم نمونے وغیرہ کی تشخیص کرتے ہیں, بیچ کا علاقہ تھوڑا ستھرا ہوتا ہے, اور بالائی حصہ جو لیب کا ہوتا ہے وہ اس لکھت پڑھت کے کام کے لیے یا ڈیٹا انٹری کے کام کے لیے مخصوص کیا جا سکتا ہے. مگر اس علاقے کے استعمال کے لیے بھی بہت ضروری ہے کہ آپ جتنے لیبارٹری کے اصول ہیں وہ فالو کریں, ان کو پوری طور پہ عمل میں لے کر آئیں, اور دیکھیں کہ آپ لیب کوٹ اپنا پہن رہے ہوں. دوسرا یہ کہ ایسی کوئی میٹنگز وہاں نہ کر رہے ہوں, کھانے پینے کی اجازت جیسے نہیں دی جاتی وہ بھی نہ کر رہے ہوں میٹنگز وغیرہ کے لیے, مگر صرف اور صرف لیب ہی کا مخصوص کام ہے اس کے متعلق آپ کام کر سکتے ہیں. ایک اور جو سوال کیا گیا ہے وہ پوچھا گیا ہے کہ مولکیولر لیب جو ہے وہ ہم ایک جو عام لیب ہے اس میں کام کر سکتے ہیں. دوبارہ یہی ہوتا ہے کہ آپ کو اپنے کام کا تجزیہ کرنا پڑے گا, جس کو دوسرے معنوں میں رسک اسیسمنٹ بھی کہتے ہیں, کہ آپ دیکھیے کہ آپ کس قسم کا کام کر رہے ہیں. آپ کا کام اگر اس قسم کا ہے کہ جو ایسے جراثیم سے کام کر رہے ہیں جو زیادہ امراض نہ پھیلاتے ہوں تو ممکن ہے کہ آپ کم کر سکتے ہیں. مگر اگر آپ وہ والا کام کر رہے ہیں, ہائی لیول کا کام کر رہے ہیں, تو آپ نہیں کر سکتے. پی سی آر کے لیے کہتے ہیں کہ پی سی آر عموماً 2 حصوں میں اس کو تقسیم کر دیا جاتا ہے. ایک وہ کام جو جراثیم سے آپ جینیٹک مٹیریل ایکسٹریکٹ کرتے ہیں, ایک وہ کام ہوتا ہے, اور دوسرا کام جو ایکچوئل جینیٹک مٹیریل پہ کام کیا جاتا ہے. تو ماسٹر مکس جو چیزیں جن میں آلودگی مکس ہو جائے, جیسے ماسٹر مکس بہت ہی اہم کام ہے, اور اس میں کسی قسم کی آلودگی نہیں آ سکتی. تو ان کے نمونوں کی جب تشخیص کرتے ہیں تب بھی کسی قسم کی آلودگی کا سامنا ہم نہیں کر سکتے. تو ایسے کام جو پری پی سی آر کہلاتے ہیں ان کو مختلف علاقوں میں تقسیم کر کے مختلف جگہوں پر کرنا چاہیے. اگر آپ کے پاس کمرے زیادہ نہیں ہیں تو ایک ہی لیبارٹری میں ہم جو ہیں الگ الگ بینچز تشخیص مقرر کر سکتے ہیں. ایسا ہے کہ پری پی سی آر کی بینچز الگ ہوں ایک الگ علاقے میں, اور جو پوسٹ پی سی آر کا کام ہے اس کی بینچز الگ ہوں. اور ہوا کا رخ کا بھی خیال کرنا چاہیے کہ جو اے لیب ہے اس سے بی لیب کی طرف ہوا آ رہی ہو, بی سے الٹی طرف ہوا نہ جا رہی ہو, جس سے آلودگی کا خدشہ پیدا ہو سکتا ہے. یہ پی سی آر کے ایک تھوڑا سا لے آؤٹ فلپ نے دکھایا ہے اور آپ کو بہت اچھی طرح سے ایکسپلین بھی کر دیا گیا ہے. اس کے علاوہ اگر ہم یہ بھی دیکھیں کہ وہ ہوا کے رخ میں نے بات کی تھی, اس کا بہت دھیان رکھیے گا کہ آپ کا ایئر فلو لیب میں ہے وہ ایک ہی ڈائریکشن میں ہو, یونی ڈائریکشنل کہلاتا ہے, کہ ایک رخ میں جا رہا ہو, جیسے اے سے بی کی طرف جا رہا ہو, الف سے بے کی طرف جا رہا ہو, بے سے الف کی طرف نہ جا رہا ہو. ایک اور سوالات کیا گیا ہے, وہ پوچھا گیا ہے کہ لیب میں جو جراثیم کا کام کیا جاتا ہے اس کے ہیزرڈز اور کیمیائی مواد جو استعمال کیا جاتا ہے اس کا ہم لیب ڈیزائن میں کس طرح سے تشخیص کریں. تو یہ بھی بہت اہم کام ہے. آپ کو پہلے دیکھنا پڑے گا کہ آیا آپ کی لیب جو ہے وہ جراثیم کی تشخیص کے لیے مقرر کی گئی ہے یا کہ کیمیائی مواد کی تشخیص کے لیے مقرر کی گئی ہے. اسی لحاظ سے لیب کا ڈیزائن جو ہے کیا جائے گا. اس کے لیے جو ہمیں اکوپمنٹس استعمال کرنے پڑتے ہیں وہ مختلف ہوتے ہیں. جراثیم کے لیے بایو سیفٹی کیبنٹ استعمال کی جاتی ہے جس میں ہیپا فلٹر ہوتا ہے, اور کیمیائی مواد کی تشخیص کے لیے ہمیں فیوم ہوڈز استعمال کیے جاتے ہیں جن کے اندر چارکول فلٹرز ہوتے ہیں. دونوں کا کام مختلف ہے. پھر بایو سیفٹی کیبنٹ جو ہے بہت مشکل ہوتا ہے کہ ایک لیبارٹری کے لیے پاکستان میں کہ ہم 2 قسم کے اتنے اہم اور اکوپمنٹس جو ہیں مہنگے اکوپمنٹ رکھ سکیں. تو یہ دیکھنا پڑے گا کہ ہمارا کام کس نوعیت کا ہے, اسی لحاظ سے آپ ڈیسائیڈ کریں گے. دوسرا سوال جو کیا گیا ہے کہ مرر جو ہے آئینہ قد آدم آئینہ لیب میں لگانے کی اجازت رہتی ہے. فلپ کا خیال ہے کہ اس کی بالکل ضرورت بھی نہیں ہے, مگر اگر آپ کو ضرورت ہے تو آپ اس کا استعمال کر سکتے ہیں. مرر میں دیکھنا ہوتا ہے کہ آپ نے جو اپنی پی پی ای پہنی ہوئی ہے وہ کس طرح سے پہنی ہوئی ہے. ضرورت نہیں ہے مگر اگر آپ چاہیں تو لگایا جا سکتا ہے. ایک اور سوال جو کیا گیا ہے وہ جراثیم کے محلول اگر گر جائے تو وہ ایک ڈبے میں ایسے سامان رکھا جاتا ہے جس کو سپل کٹ کہتے ہیں, اس سے وہ صفائی کی جاتی ہے. تو یہ سپل کٹ جو ہے یہ کتنی اہم ہے, اس کے لیے بھی آپ کو رسک اسیسمنٹ کرنا پڑے گا کہ اپنی سپل کٹ کے اندر کس قسم کا سامان رکھنا ہے. آیا کہ آپ کیمیائی مواد کا کام کر رہے ہیں, آیا کہ آپ جراثیم کی مواد کا کام کر رہے ہیں, تو اس لحاظ سے آپ کی سپل کٹ تشخیص کی جائے گی. سپل کٹ کہاں رکھنی چاہیے؟ سپل کٹ کو اینٹی روم میں رکھنی چاہیے یا سپل کٹ اسی جگہ پہ رکھنی چاہیے جہاں کام کر رہے ہیں. بہت ضروری ہے کہ آپ یہ دیکھیں کہ جب کوئی جراثیم کے محلول کا آپ کوئی گر گیا ہے جراثیم کا محلول اور اس کو صاف کرنا ہے تو آپ کو سب سے پہلا قدم یہ لینا پڑتا ہے کہ وہ آپ کمرہ خالی کر لیں, اس لیے کہ جو بخارات ہیں اس محلول کے محلق ہیں فضا میں وہ کم سے کم 20 یا 30 منٹ کے اندر یا 50 یا اس سے زیادہ کے اندر زمین کی سطح پر جم جاتے ہیں. تو آپ کو سب سے پہلے جگہ خالی کرنی پڑتی ہے. تو اس لیے بہت ضروری ہے کہ سپل کٹ آپ کی جو ہو اس کمرے میں نہ ہو, بلکہ بہتر یہ ہے کہ اینٹی روم میں رکھی جائے یا کسی کوریڈور میں لیب کے رکھی جائے. کوریڈور میں رکھنے کا یہ بھی بہت اچھا آرام ہوتا ہے کہ بہت ساری لیبیں اس کو استعمال کر سکتی ہیں. اینٹی روم بالخصوص صرف اسی کام کے لیے استعمال کیا جائے جس کے لیے وہ مقرر ہے. وہ آپ کی پی پی پہننے کے لیے, ہاتھ دھونے کے لیے, اس کو ایک سٹوریج یا چیزیں جمع کرنے کا علاقہ نہ بنایا جائے. شکریہ.
Thank you very much, Shaila. So the second part is about the technical aspects related to ventilation and air conditioning. This is a more technical part, and so again for this part, you should have your requirements, you should define things, you should discuss things, but of course you should work with competent engineers and technical persons, including your maintenance people that have most of the time a very practical view of the situation. So the first question was kind of preliminary. Is humidity dependent on the nature of work? Actually, in general, humidity is mostly related to climate conditions, and because of that, excessive humidity may be an issue in many parts of Pakistan and in many Pakistani labs. There is no risk of creating a lot of humidity during the scientific process, technical process, because we don't use large volumes of liquids, and it's we don't kind of boil it or anything like that. Actually, the main exception is autoclaving, and we use an autoclave for media and material preparation, mostly sterilization. Then we can also use autoclaves for decontamination, for waste treatment, and so it can be in a decontamination room, and also autoclave is something placed in the labs, mostly for sterilization purpose. So you see that autoclave may be present in lab, and in that case it will create heat, and also humidity, and that is prejudicial to the comfort, but also to the risk of contamination. So a first recommendation would be to first avoid installing an autoclave in the lab itself, especially not in labs where there are some diagnostic or research activities. So it would be either in a sterilization room or in a decontamination room, but not in the lab. Okay, and so it should be in a dedicated room if possible, so that you don't create a humidity and heat problem inside of your lab. Okay. Another question was related to that, said which ventilation technique should we use for an autoclave? Actually, autoclaves release a lot of steam when they are opened at the end of the treatment cycle. So when you open the door after the treatment cycle, in that case, they release a lot of steam, which means a lot of heat and humidity. And this is really an issue if it's present in a lab. So with respect to ventilation, there are different possible solutions. If it's a decontamination autoclave, it could be in a rather open environment, which is not very ventilated. Of course, if it's high risk biological waste, and if you don't want to take any risk of spreading the material, in that case, that solution is not acceptable. It's not good for a sterilization autoclave either, because you want to have your material sterile when you get out of the autoclave. But in other situations, you can have it in a quite open environment so that it's naturally ventilated. That's the easiest and most affordable solution. If it cannot be done, in that case, the best practice, and that's easy to do, is to install a canopy with an exhaust fan above the door of the autoclave. A canopy is a kind of hood, like it is like on the picture there, and with a duct that takes the air toward the outside. In that case, you would eliminate the steam and thus most humidity and heat immediately from the start, close to the source. Here are a few pictures of different types of autoclaves, each of them with a canopy above it. Okay, that canopy could possibly be homemade. It does not have extremely fancy. It just had to collect the air that is exhausted with a fan. The fan itself should be powerful enough, and certainly much more powerful than the fan of a biosafety cabinet, for instance. And it should exhaust the steam outside of the building. It should not exhaust it in the next room. That would be kind of also prejudicial. So outside of the building. A good thing also is that it doesn't work all the time, because you will exhaust a lot of air. So it will cost a lot of energy. And it could only be activated either automatically or manually when you need it. That means at the end of a cycle. So when after a cycle of an autoclave, let's say that lasts about 2 hours, at the end of the cycle, you turn off the fan and then you open the door. And in that case, you would get rid of the steam immediately. The next question is, does an exhaust fan in the presence of a class 2 biosafety cabinet create disturbance of the airflow inside of the BSC? I termed that as a question, but actually it was a kind of assertion from one of you. Clearly, the answer is absolutely not. This is a common misconception, and even some engineers don't see that very clearly. But the real thing is that if any device sucks the air, it has no impact on airflows and no impact on the functioning of a class 2 biosafety cabinet. On the other hand, if it's a device that pushes the air towards the biosafety cabinet, in that case, it's likely to have a significant impact. Okay, so if you take the air away from the cabinet, no impact. If you push the air towards the cabinet, in that case, you will have an impact. This is very clear, and a good way to remember that is to think about a candle. You can only extinguish a candle if you blow, if you push it somewhere towards the candle. If you suck the air in that case, it's impossible to extinguish a candle. So try to remember that image of you being extinguishing a candle. Only the air that is pushed will disturb the biosafety cabinet, not the air that is sucked. So the best practice actually would really be to locate the exhaust fan above or very close to the biosafety cabinet. Why? Because it's a high risk zone. So if you have a spill, you will evacuate those infectious materials as soon as aerosols as soon as possible. Also because you will evacuate the heat which is produced by a fan of the biosafety cabinet, and a biosafety cabinet produces quite a lot of heat, and you will also evacuate it immediately. So clearly, the best location of an exhaust fan or an exhaust grill of an HVAC system is above a biosafety cabinet. You could also even use a canopy, like the one that we put above an autoclave. The next question is really an important one, because it's basically, the question is how to manage the issue of the cost of rooming cool air with ventilation. And the actual questions are there. So labs in warm and resource-limited countries like Pakistan can use exhaust fans. However, when we use it is at the cost of removing the cool air in the lab, and then it's needed because of PPE and so on that are used in the lab. Since HVAC systems, I told you, are very expensive, what could be the possible solution for that? And the other question was also kind of opposing exhaust fan versus air conditioning labs. But actually, with HVAC system, the issue is exactly the same. You provide some air in one way and you extract it the other way. So you should avoid to eliminate too much of that air which is cooled at high cost. Okay? It's a real issue and a very important issue with respect to cost and sustainability. Some ventilation is needed, it's very clear, and you saw that during the first sessions. And in a country like Pakistan, cooling is also needed. And in addition to the natural heat, the one that is produced by climate, you also have many pieces of equipment in the lab that generate high heat loads, like fridges, freezers, incubators, biosafety cabinets, autoclaves, computers, and so on. So you really need cooling, and so you need both ventilation and cooling. Cooling and also the humidification of the air to suitable condition is very costly. It's the cost of equipment, but it's also the cost of energy, which can be very high, and also the maintenance cost. And of course, if you cool down the air at high cost and you ventilate, you will evacuate that air. So that I only explain, it's not the answer, I only explain in some way the question and what it is such an important question in laboratory design. Actually, there is no unique solution to deal with that. But the issue has to be really dealt and studied by competent architects and engineers who are familiar with the situation in Pakistan or in similar countries. So it has to be taken very seriously and studied by competent people. Some considerations. The first thing to do, which is very important, is the architecture and also the construction means. These might have a major impact on the heat within a building. So the most heat you have inside of the building, the most cooling you will be needed. And if you prevent that heat to accumulate within the building by good construction techniques, for instance, you will need much less cooling. And this is not a job for laboratory people. It's really a work for architects and engineers. And those factors are, for instance, the orientation of the building. The fact that you could limit the large openings, large windows to the not having them to the south, but right to the north of the building. Also, the structure of the walls and openings. The walls should be kind of in two layers with some ventilation in between. Also, more energy efficient materials and systems could be used. You could also use passive ventilation in vertical space like stairways to just cool down the building. And also, if you have some very sensitive labs that need a lot of cooling, you could also protect them in putting them in the middle of the building. So clearly, sustainable design and construction principles should be used. Other consideration, the first thing is that you could possibly separate and better isolate and ventilate labs and rooms which need the most, because there is a lot of heat which is generated. That means autoclave rooms, freezer storage rooms. So those should be separated from the labs and should be better isolated and also very well ventilated. Also, you should define the size of the air conditioning systems. It could be, it's true for HVAC, but it's also true for the fans. And that should be calculated when considering the heat generation in each lab. And so you should keep in mind the heat from the climate, but also from the equipment, from the people, and so on. Of course, this is not something that we can do as scientists, but this has to be done by engineers on the basis of the information you will provide them with. The ventilation system itself, so the fan, should also be dimensioned the good way. And for instance, you should not use very high air change rates. Like in the US, for instance, the kind of standard for air change rates in a lab is 20 changes of air per hour. This is huge. In Europe, we use like 6, 8, 12, but even 8 in most situations is not needed. So you could ventilate at a very low rate. And if your fan is well located, you won't need to change the air very often at a very high rate. So the location of the fan and the supply air is more important than the number of air change rates. Another trick is that you should evacuate heat from the main sources as close as possible to the source. And I gave the example of the biosafety cabinet and the autoclave. And also you should install the air conditioning device, so the system that is cooling the air, at the opposite end versus the ventilation system. So you should extract the air as far as possible as the place where the cool air is generated. Still, with respect to this issue, there are a few general things that we need to consider. A very important thing, and is to adopt sustainable design and construction principles. And actually, many traditional techniques in warm countries can be used for constructions of labs. So don't consider that because it is something which is kind of trivial in your country, that is quite usual because it's traditional, that it's not good. Most of the time, there are very good options there. Of course, you can also join some new techniques, but only if they are affordable and sustainable. But for instance, having a kind of vertical ventilation in the middle of a building to cool down the building itself could be a good and affordable sustainable system. Another general tip in that respect, and in many respects I would say, is that we should not use Western standards or copy-paste Western solutions, at least not without considering their impact on cost and sustainability. And this is very important. Why? Because most Western countries don't have the same issues than in Pakistan and not the same climate as in Pakistan with respect to ventilation. Then the technical and financial capabilities in the West are totally different from what you can find in Pakistan. Also, the way we deal with cost and sustainability is totally different. And so copy-pasting Western solution is not the good way to do. You should only consider possible good options among those if they appear sustainable and if you can really afford it in your country. And so it's time for some conclusions, mainly based on that. And I took those from a very good architect from the US, who is the leader of training on guidelines for laboratory design, Mrs. Tannenbaum. And she considered that there are 3 keys to sustainable laboratory design. The first thing is to involve a wide range of stakeholders in designing the lab. Project management is extremely important when you want to design and construct and build a lab. We did not deal with this in this training, but perhaps in the future we should have that opportunity. But it is really something which is very important. I've seen projects really going down and not being achieved because of problem of project management. I've seen many laboratories that were built, but finally were not operational because of problem of project management. So it's really important to gather all the stakeholders and having a good project organization. A second one, second key is to that we must ensure that the standards that we adopt for sustainability fit our lives. I mean, respond to our needs. So they need to do those options, the possible options with respect to sustainability, need to respond to our operational needs, protection needs, so based on risk assessment, and they need to be based on locally available capabilities. If each time you have an issue with your facility, with your air conditioning system or so, you have to call in people from abroad, it will not be sustainable. The third one is to balance sustainability with health and safety. You will be surprised that I give that as a conclusion, but I think it's a very good one. Of course, when we deal with biosafety, we think about health and safety. But if we kind of ignore sustainability aspect, if we adopt solutions which are not sustainable, actually will compromise safe operation in the future. So this is why we should not only look at biosafety, at the safety of the people or safety of the environment and the population, but we should also look at the sustainability issue. Otherwise, in the long term, we'll compromise safe operations. I think it's important. I know that most of the workshop was on quite more or less technical issues, but I think it's very important to also have that kind of considerations in mind when we deal with design and construction. So now it's time for also a summary of this last part. Please, Ms. Shaila.
आप सब लोग स्क्रीन देख सकते हैं मेरी? हेलो, स्क्रीन नजर आ रही है मेरी?
Yes, we see.
Yes, we can see.
चलिए, पहला सवाल जो किया गया था और वो है जो सेकंड पोर्शन था हमारा इस लैब डिजाइन वेबिनार का, उसमें जो सेकंड सेक्शन का पहला सवाल जो है वो है जो नमी लैब के अंदर होती है कि काम के अंदर, फिजा के अंदर जो नमी है, काम बहुत ज्यादा डिपेंडेंट करता है नमी कितनी मुकर्रर की जाए लैब के अंदर। लैब एक खास एयर सर्कुलेशन और नमी के ऊपर काम करती है। तो नमी जो है वो नॉर्मली जो फिजा है, किस किस्म का मौसम है आपका, गर्मियों में खासकर जबकि गर्मी बहुत होती है तो लेबोरेटरीज में पाकिस्तान में नमी ज्यादा हो जाती है। तो इसके लिए बाज ऐसे जो हम इस्तेमाल करते हैं इक्विपमेंट, वो एग्जांपल ऑटोक्लेव इस्तेमाल करते हैं जो मीडिया प्रेपरेशन के लिए करता है, यूज होता है। इसके अंदर स्टीम प्रोडक्शन होती है, भाप के बुखारात बहुत ज्यादा होते हैं और बहुत ज्यादा हीट प्रोडक्शन हो रही होती है, हीट बहुत इस्तेमाल होती है जिसकी वजह से कमरे में नमी पैदा हो जाती है। तो बेहतर होता है कि आप इस किस्म के जब काम करें तो उसको देखिए कि ये इस इक्विपमेंट्स हैं जिसमें आप अपने लैब वेयर धो रहे होते हैं, साफ करते हैं, हीट स्टरलाइजेशन यूनिट्स होते हैं और ऑटोक्लेव्स जो हैं इनको कहाँ पे रखें, इन चीजों को किस कमरे में रखा जाएगा, ये नमी के दबाव को कम करेगा। और जो क्योंकि ये बहुत ज्यादा स्टीम प्रोड्यूस करता है और स्टीम का प्रेशर बहुत ज्यादा होता है, इसलिए ये हवा में मुहल्लक हो जाता है और उसकी वजह से काफी टेम्परेचर जो है वो हाई हो जाता है। तो ऑटोक्लेव्स के ऊपर बहुत अच्छा बताया है फिलिप ने कि हम लोग एक किस्म का कैनोपी रख सकते हैं जो कि जितनी भी स्टीम प्रोडक्शन या भाप के जो बुखारात हैं वो सारा ट्रैप हो जाएगा उस कैनोपी में और डायरेक्टली सीलिंग से अटैच होगा। तो ये जितना टेम्परेचर, हाई टेम्परेचर है और बुखारात जो हैं उसके, ये सारे ट्रैप करके बाहर ले जाएगा। تو بہتر ہے کہ ایسی جگہ پہ جہاں آٹوکلیو رکھیں آپ. اگر کمرے میں رکھا جا رہا ہے اور آپ کسی الگ کمرے میں نہیں رکھ سکتے ہیں تو اس کے اوپر کینوپی ضرور لگائیے گا. دوسرا سوال جو کیا گیا ہے وہ ایگزاسٹ فین، کہ کئی جگہ یہ کہا جاتا ہے کہ جس کمرے میں بایو سیفٹی کیبنٹ رکھی جائے وہاں پہ ہوا کا رخ ڈسٹرب نہیں ہونا چاہیے. تو یہ فلیپ کا کہنا ہے کہ یہ بالکل غلط کہنا ہے، کیونکہ یہ بایو سیفٹی کیبنٹ جو ہے ایک یونی ڈائریکشنل، ایک سک کرتی ہے ہوا باہر کی اندر کی طرف. تو اگر آپ سک کر رہی ہے تو اس میں اگر آپ کوئی اور آلات لگا بھی دیں لیب کے اندر تو وہ ہوا کے رخ کو بدل نہیں سکتی. وہ اتنی پریشر پہ کام کر رہی ہوتی ہے کیبنٹ کہ وہ ایک ہی ڈائریکشن میں ہوا چل رہی ہوتی ہے. تو یہ کہنا کہ بھئی یہ کسی طرح سے نقصان پہنچائے گا بایو سیفٹی کیبنٹ کو، یہ غلط ہے. تو یہ بتایا جا رہا ہے کہ اگر ہم لوگ ایگزاسٹ فین بالکل بایو سیفٹی کیبنٹ کی اوپر لگا دیں، سیلنگ کے اوپر، تو اس کا جو ہوا کا رخ ہوتا ہے، جو پروڈیوس ہو رہا ہوتا ہے ہوا میں، ہوا کی ڈائریکشن جو بنا رہا ہوتا ہے یہ لیب میں، اس سے بایو سیفٹی کیبنٹ کی جو اپنا کام کرنے کا طریقہ ہے اس پہ کوئی اثر نہیں ہوتا. اب دوسرا جو بہت اہم سوال کیا گیا ہے اس سیکشن میں وہ یہ ہے کہ بھئی کیونکہ ہمارے ملک میں گرمی زیادہ ہوتی ہے اور خاص کر گرمیوں کے وقت میں موسم کے اندر ایسا رہتا ہے کہ لیب میں اگر آپ ایک خاص ہوا نہ مینٹین کر سکیں اور ایئر کنڈیشن بھی چلائیں اور ایگزاسٹ بھی کر رہے ہیں اسی وقت، تو اس میں بہت ساری جو ٹھنڈی ہوا ہے وہ ایگزاسٹ کے ساتھ باہر چلی جاتی ہے، جس کا نقصان زیادہ ہو جاتا ہے اگر ہم نے ایگزاسٹ فین لگایا ہوا ہے. بہت ضروری ہے کہ ہمیں ہیوک سسٹم لگانا پڑے گا، اور ہیوک سسٹم کیونکہ بہت مہنگا ہوتا ہے اور اس کے استعمال کے لیے پیسے زیادہ لگتے ہیں، تو یہ بھی ایک بحث ہے کہ آپ ایگزاسٹ لگائیے یا ایئر کنڈیشننگ سسٹم لگائیں. دونوں چیزیں اپنی جگہ اہم ہیں. تو یہ پوچھا جا رہا ہے کہ بھئی کس طرح سے ہم ٹھنڈی ہوا باہر نہ نکل جائے لیب میں سے، اور اس کا نقصان ہوگا اگر ٹھنڈی ہوا باہر نکل جائے گی تو آپ کا جو ایئر کنڈیشننگ یونٹ ہے وہ بہت زیادہ کام کرے گا اور کاسٹ زیادہ پڑ جائے گی آپ کو. لیب میں نارملی پاکستان میں کام کرتے وقت جو لوگ اگر بہت ہائی لیول کا کام کر رہے ہیں تو ان کو پی پی پہننی پڑتی ہے، لیب کوٹس، اس کے ساتھ ہولڈ، اور اگر ٹائیویک سوٹس بھی پہن رہے ہیں تو اس کیس کے اندر لیب میں اگر پراپر بہت مخصوص قسم کی ہوا کا دباؤ نہ مینٹین کیا گیا اور وہ ایئر کنڈیشننگ اور ٹھنڈی ہوا برابر طور پہ مینٹین نہ کی گئی تو اس کی وجہ سے لوگوں کے کام کرنے میں دشواری ہو سکتی ہے. اور جتنے آپ لوگ ہم لوگ اکوپمنٹ رکھتے ہیں لیب میں وہ بذات خود بھی ایک ہیٹ نکال رہی ہوتی ہیں اپنے فنکشن کے اندر، تو وہ ہیٹ بھی ایڈ ہو جاتی ہے کمرے میں. تو اس کے لیے بھی بہت ضروری ہے کہ ہم یہ دیکھیں کہ ان چیزوں کو کم کیسے کر سکتے ہیں. یہ پوچھا گیا ہے کہ بھئی یہ کیسے مینیج کیا جا سکتا ہے کہ نقصان کو ہم متبادل کیسے کریں. تو یہ جب لیب ڈیزائن کی جائے اس وقت ان چیزوں کو دیکھا جائے، اور یہ زیادہ سے زیادہ آرکیٹیکچرل ایشوز ہوتے ہیں جن کو دیکھیں کہ لیب کا آپ بنا رہے ہیں تو لیب ٹیبس کے کمرے جو ہیں وہ تیز دھوپ کے رخ پہ نہ رکھے جائیں تاکہ باہر کی گرمائش جو ہے وہ لیب کو زیادہ گرم نہ کر دے، اور آپ جو ہوا کا دباؤ ہے وہ سٹیئر کیسز سے ہوتا ہوا اوپر آ رہا ہو، کوئی ڈائریکشنل ایئر فلو جو ہے وہ مقرر کی جائے اس طرح سے کہ کمرے بھی ٹھنڈے ہوں اور ساتھ ساتھ دباؤ آپ کے ہوادار کمرے بھی رہیں. اور ایسے آلات جو ہم استعمال کر رہے ہوں لیب میں جو سٹیم یا ہیٹ بہت زیادہ نکال رہی ہوں، ان کو یا تو الگ کسی کمرے میں رکھیں یا جیسے فلیپ نے بتایا ہے ان کے اوپر کینوپی انسٹال کی جائے. یہ چیزیں جو ہیں ان چیزوں کا استعمال اگر ہم دھیان سے کریں گے تو ہمارے کمرے ٹھنڈے رہیں گے. دوسرا یہ پوچھا گیا، یہی سوال جو ہے بہت ہی اہم ہے اور اس میں فلیپ کا یہی کہنا ہے کہ بھئی کاسٹ بہت زیادہ لگتی ہے ہیوک سسٹم میں. آپ جب بھی کبھی لیب بنائیں تو بہتر ہے کہ آپ ان تمام چیزوں کی طرف پوری طرح سے دھیان دیں کہ آپ کا اپنا لیب کا جو ہے اس میں ہوا کا رخ کس طرف سے آ رہا ہے، ایگزاسٹ کس طرف سے ہو رہا ہے، کمرے کا جو ایئر پریشر ہے، ایئر ایکسچینج ہے وہ کتنا مقرر، جیسے 20 ایئر ایکسچینج पर आवर मेंटेन करना चाहिए। और अगर ये ना हम मुकर्रर कर सके तो फिर हवा आपके उसमें नमी प्रोड्यूस हो जाएगी कमरे के अंदर और हवा का दबाव भी ज्यादा बढ़ जाएगा। तो ये तमाम चीजें हमें ध्यान से करनी चाहिए कि हम उस वक्त जब लैब बना रहे हो इन चीजों को ध्यान दें कि कमरे किस रुख पे, हवा के किस रुख पे हैं, साउथ की तरफ है, वेस्ट की तरफ है, जुनूब है, शुमाल है, या के धूप के रुख की तरफ ना हो। और कंक्लूजन में बहुत अच्छी तरह से फिलिप ने खुद बहुत एक्सप्लेन कर दिया इन चीजों को, तो मेरे इतने बताने की जरूरत नहीं है। और यहाँ पे हमारा ये सेशन खत्म होता है।
Thank you very much, Shahida. I think it's now time to really conclude this this webinar. I really want to thank the people who helped with the translation. Shahida today, but before that, we also had the help of Zara and also Khalid. So thank you to those. Thank you also for the PETSA team for helping with the different aspects, including the fact that today had some laptop problems, but anyways, it's solved. And thank you to all of you for your participation. And I hope that you will keep at least the main ideas of this workshop, and that it will help you to improve the situation in your lab, and perhaps someday to really participate to the design and construction of a new lab. So thank you very much.